Hi and good day. I am Fatika Arbahia E25A3966 as a leader for our group assignment. And today, we will present our assignment on subject biology and molecule. The topic that we chose is Oryza sativa, or commonly says as rice. We will follow exactly the 3 required points, which is chromosome number, gene number, and discussion on genes with application. Starting with point 1, number of chromosomes. Rice is a diploid organism, meaning its cells contain 2 sets of chromosomes. According to cytogenetic data, rice has a total of 24 chromosomes arranged in 12 pairs. In scientific terms, we write this as 2n equals to 24. It has one of the smallest genome size among cereal crops, about 430 million base pairs. This small size is why rice became the first crop plant to be fully sequenced, and it is used as a model organism to study genetics for all other crops, like this. Here, you can see the picture showing the karyotype or arrangement of rice chromosomes. That's all from me. I will pass to the next person.
Thank you. I am Muhammad Nabil Farhan bin Muhammad Fazil, presenting point number 2. Number of genes supported by published journal articles. Genes are segments of DNA that carry instructions for life process. Based on complete genome sequencing, Oryza sativa contains approximately 37,000 to 39,000 genes. Interestingly, rice has more genes than humans because plants need estrogens to adapt to environment, weather, and disease since they cannot move. To support this data, we refer to a published journal, International Rice Genome Sequencing Project 2005. It is a landmark research published in the prestigious journal Nature. An international team recorded the entire rice DNA and confirmed there are 37,545 to 39,045 protein coding genes. This is the official reference used worldwide today. These genes control everything from grain size, aroma, heat, to disease resistance.
Now, we move to point 3, the main part of this assignment. We will discuss one specific gene, its protein, and how it is used in agriculture, supported by case studies and articles plus critical analysis. What is its function? This gene was originally found in white rice species. The protein is produced acts like a security sensor located on the cell surface. When harmful bacteria called Xanthomonas aureus attack the plant, this protein detects the bacteria immediately, send chemical signal inside the cell, and activate the plant's defense system. This stops the disease called bacterial blight, which can destroy 50% of crops. Here is the diagram showing the protein structure and how it works. Because of this powerful ability, scientists took this gene and applied it in agriculture. Nabil will explain the exact method used.
Thank you. I am Muhammad Nabil Amshah bin Muaz Nizam, and I will explain the application and the detailed method as required in the assignment. Application: improvement of crop production in agriculture. The method name is Marker Assisted Selection, which is MAS. Before this method, breeding took 10 to 12 years. Now it is much faster. Let me explain step by step. The first step is scientists identified the exact location of SA21 on chromosome 11. They created a DNA marker, a unique genetic tag that exists only if SA21 is present. The second step is they cross wild rice with SA21 with good quality rice varieties that farmers like but are easily sick. The third step is the most important part. Instead of planting and waiting, they take a tiny leaf sample, extract DNA, and test in the lab. If the marker shows up, we know immediately the plant has the resistant gene. No need to infect it. And the fourth and fifth step, they breed the selected plants to keep all good qualities plus the new resistant. This diagram shows the process clearly. This method combines molecular biology with traditional breeding. Now, Wada will show the published article, case study, and result. That's all.
I am Nul Sore Mitikasi, E25A3973. I present the case study and published article regarding this method as required. This famous study in Science Journal was the first to successfully clone and characterize the XA21 gene. It proved that this single can give wide resistance against almost all types of bacterial blight. Case study result using the marker assistant selection method we explained. Scientists developed a new rice variety named IRBB21. Later, this was transferred into local varieties in Malaysia, China, India, and Philippines. The comparison photo on the left, rice without XA21, leaf a dry dead no herbs. On the right, rice with XA21, green, healthy, full grains, even when infected. Reduce chemical pesticide used by 40%, safe for environment, stable for supply. This is real proof of how genetic knowledge solves problems. Finally, next person will give the critical analysis as required.
Thank you. I am Muhammad Nasifan bin Muhammad Nazri, E25A3900. I will complete point 3 with critical analysis. That will explaining how this research improves genetics, molecular biology, and agriculture. Firstly, improvement in genetics. Before XA21, we did know how plants defend themselves. This discovery proved plants have immune system similar to animals. It established the concept of resistant genes and changed our basic understanding of plant genetics forever. After that, improvement in molecular biology. The success of marker-assisted selection proved that we can select traits using DNA, not just by looking at the plant. This method becomes a standard tool used for all crops worldwide today. It's linked theory to practical application. Next, for impact and limitation. For the pros, faster breeding, accurate, safe, no foreign DNA introduced. For the limitation needs lab cost and skilled worker. Also, bacteria evolve, new races may overcome resistance. So research must continue.